Hi everyone, my name is Günel Əyyubova. I am senior lecturer of Histology department, and today's our lecture is about the organelles of the cell, that is the centrosome, mitochondria, endoplasmic reticulum, Golgi complex, endosomes, lysosomes, peroxisomes, and proteasomes. The objective is to get knowledge about the cellular compartments, their functional relationships, their appearance under the light and electron microscope. Learning goals, these are characteristics of centrosomes, centrioles, and other organelles. References, these are the literature books, textbooks which you may use during your studying. First of the organelles which are studying, that is the centrosome. Centrosome is a region of the cell that is in the vicinity of the nucleus and that houses the centrioles. We are looking at the centrioles, and also around the centrioles we have pericentriolar matrix that include gamma-tubulin ring complex molecules. The centrioles, they are joined to each other via connecting fibers in some species. And from these centrioles, one of them is mature centriole, which has subdistal appendages, these are, and also satellites here. The immature centriole have not those appendages. And in the center of the centrioles, we have protein centrin. And then the centrioles we can see here in the electron microscope representation. As we can see, two centrioles, they are located at the right angle to each other, and at the onset of mitosis during the cell's interphase, each half having a duplicated centriole pair. Under the electron microscope, we are looking here at the longitudinal section and the cross section of the centrioles. And here the centriole structure. Actually, each of the centriole is composed of the 9 microtubular triplets. 9 triplets form the walls of the cylinder. And here we are also looking at the centriole structure, which contain 9 triplets of microtubules. And we have to know that in the pericentriolar matrix we have these gamma-tubulin ring complex molecules, and these gamma-tubulin molecules act as nucleation sites for microtubules, which are long, straight, rigid, and hollow-like cylindrical structures, and centrosome, so it's considered to be the microtubule organizing center, because in here start all of the microtubules of the cell. It is here taking place assembly of the tubulins, which further form the microtubules of the cell. Centrioles, they are small and cylindrical structures, as we are looking here, composed of 9 triplet microtubules, and they constitute the core of the microtubule organizing center, or the centrosome. So usually the centrioles are paired structures arranged perpendicular to each other and are located in the microtubule organizing center and in the vicinity of the Golgi apparatus also. Here we can see the centrosome assists in the formation and organization of the microtubules, as well as in the self-duplication before cell division. Centrioles are composed of specific arrangement of microtubules, and here microtubule triplets, they consist of each of the microtubule triplet. Here it consists of the microtubules which signed A, B, and C. The complete microtubule A, it positioned closest to the center of the cylinder, and the microtubule C is the farthest away. Microtubule A composed of 13 protofilaments. It has complete structure, but the B and C microtubules, they have incomplete structure because they share protofilaments with the neighbor ones. That's why they include just 10 to 11 protofilaments. Adjacent triplets are connected to each other by a fibrous substance which extend from microtubule A to microtubule C. Each triplet is arranged so that it forms an oblique angle with the adjacent triplet, as we are looking here, and a straight angle with the fifth triplet. Centrioles function in the formation of the centrosome and during mitotic activity, and they are responsible for the formation of the spindle apparatus. We are looking here the spindle apparatus that extend from one centriole to the next centriole, and these are microtubules of spindle apparatus. Then in the center we are looking at the chromosomes. Here also they located in the equatorial plate. It is a metaphase of cell division. Additionally, centrioles are the basal bodies that guide the formation of the cilia and flagella. And during the, here we are looking at the centrioles and microtubules that radiate from the pericentriolar matrix. During the S phase of the cell cycle, each centriole of the pair replicates and forming a procentriole in some unknown manner at the 90 degrees to itself. This procentriole, which is yellow colored here, initially possess no microtubules, but tubulin molecules begin to polymerize closest to the parent centriole, with the plus end growing away from the parent. The axial replication of the centriole requires the presence of gametubulin rings structures that do not become part but serve to direct the elongation of the forming microtubules by occupying the forming plus and minus ends. Additionally, gamma tubulins related to the alpha and beta tubulin superfamily also required to form the triplet structure of the new microtubule arrays. So before cell division, before mitosis, in the cell we will have two centrosomes, each of which include two centrioles and pericentriolar matrix. That then one of them will move to one pole and other one to the opposite poles. By that way, that mitotic spindle apparatus will be formed. The next organelle which we have to know, that is the mitochondria. They are round and or slightly elongated structures, and they are mostly numerous in the cytoplasmic regions with high energy demands, because mitochondria, they are powerhouses of the cells. That's why the cells which require more amount of energy because of their movement or other need, they have more amount of mitochondria. And so in cells stained with hematoxylin eosin, the mitochondria appear throughout the cytoplasm as numerous eosinophilic structures, as this one. But in cultured endothelial cells appeared as elongated structures, yellow or orange, parallel along the microtubules. We have to know that mitochondria, they are flexible and rod-shaped organelles. They have large number, most animals have more large number of mitochondria, because via oxidative phosphorylation they produce ATP, a stable storage form of energy that can be used by cell for its various energy requiring activities. Each mitochondrion possesses a smooth outer membrane and folded inner membrane. The folds of the inner membrane, they are known as cristae. They increase the surface area of the membrane. The number of cristae, they related directly to the energy requirement of the cell. Between the membranes there is an intermembranous space. Intermembranous space here and here, this one, and also the large space enclosed by the inner membrane. It is termed the matrix space. It is here, the matrix space. Outer mitochondrial membrane and intermembranous space here. They possess large number of porins in the outer membrane. Multipass transmembrane proteins. Each porin forms large aqueous channel. So in the outer membrane we have large channels that permit entrance of substances which are less in the as large as 10 kilodalton, and they may pass. But because this membrane is relatively permeable to small molecules, including proteins, the contents of the intermembranous space is resemble the cytosol. Additional proteins located in the outer membrane are responsible for the formation of the mitochondrial lipids. Now let's look at the inner mitochondrial membrane, which has more complicated structure. In the inner mitochondrial membrane, it is impermeable for much of substances. It's related to the presence here the cardiolipin molecules, which are types of phospholipids that include 4 rather than the usual 2 fatty acid chains. And in some regions, the outer and inner mitochondrial membranes, they contact each other. They contact each other at some regions, and in the contact sites, these contact sites act as pathways for proteins and small molecules to enter and leave the matrix space. The contact sites are composed of the carrier proteins and different for the transport and the regulatory proteins for the recognition of markers. And so there's also 5 protein complexes which together called electron transport chain molecules. And also we have to know that there are also transporters for calcium ions and transporters for the pyruvate together with the hydrogens, then pyruvate, the hydrogens together with phosphate groups. So these are specialized transporters. They present in inner mitochondrial membrane because it's impermeable for many substances. That's why the substances to be transported, they will be that process will be possible by the way just of the specialized carrier proteins. So now we will look at the matrix of the mitochondria. Actually, the matrix space is filled with a dense fluid composed of at least 50% of proteins that accounts for its viscosity. Much of the protein components of the matrix are enzymes responsible for the degradation of fatty acids and pyruvate to the metabolic intermediate acetyl coenzyme A and the subsequent oxidation of the intermediate in the 3-carboxylic acid Krebs cycle. Mitochondria also have ribosomes, tRNA, messenger RNA, and it also has dense spherical matrix granules. The matrix granules, they contain calcium and magnesium ions. So the matrix also, and here we also have the mitochondrial DNA, which have circular structure, and mitochondrial DNA actually it is the code for the just 13 proteins of the mitochondria. And so okay, so that is the just 13 proteins, and the proteins which are participate in the process of Krebs cycle, those are encoded by the mitochondrial DNA. The other proteins of the mitochondria, they are encoded by the nuclear DNA. And also we have to know that mitochondria are self-replicating in that they are generated from pre-existing mitochondria. And these organelles enlarge in size, replicate the DNA, and undergo fission. The average lifespan of mitochondria is 10 days. And here we are looking at the mitochondria crista or tubular crista, which is characteristic for the steroid hormone synthesizing cells. For example, in the adrenal cortex, we have mitochondria with tubular crista. And but this one is shelf-like crista here, and that is the tubular crista. So we can distinguish these mitochondrial crista shapes in different types of cells. And now we have to talk about the processes which are taking place in the mitochondria. Here in the mitochondrial inner membrane, I mentioned that there are specialized protein complexes which together called electron transport chain. Also here we have ATP synthase molecules. These are globular units on short stalks. They are needed for generation of ATP molecules. Metabolites that reach in the energy, they need to undergo degradation to release energy. We know that the substances which reach in energy, they are glucose, carbohydrates, and fatty acids. The initial step of degradation of glucose, it's taking place inside of the inside of the cytoplasm, and there the glucose subdivides into two pyruvate molecules, and they're released to ATP molecules. Then pyruvate and fatty acids, they enter mitochondria via membrane porins. Just I mentioned that in outer membrane we have porins, large pores, through which these substances may enter the intermembranous space. And from there into the matrix, they carried by specialized carrier proteins, pyruvate, and fatty acids enter the matrix of the mitochondria. Here, these substances undergo some change, and first of all, they are converted to acetyl coenzyme A that enters the citric acid cycle, Krebs cycle. And during this citric acid cycle, there are produced ATP molecules and also some intermediate substances like NADH and FADH, which are major sources of electrons for electron transport chain. And these electrons then will be transported through the electron transport chain from first complex to second, then to third, fourth. When the electrons are transported, the hydrogens from the matrix, each step they are carried to the intermembranous space. By that way, there is created a gradient of concentration, of the protons in both sides of the inner mitochondrial membrane. Then, when the hydrogens return to the matrix, they return just via one way, that is the ATP synthase. And so here we can see the mitochondria. This is circular DNA of the mitochondria, and that is outer membrane and the inner mitochondrial membrane. These are various transporter molecules here, carrier proteins, and then here we are looking at the protein complexes of the electron transport chain in this illustration. And here we can see that hydrogen protons are carried to the intermembranous space, and by this way they create gradient of proton gradient, the gradient of concentration of protons. Their amount increases in intermembranous space. Then when they return to the matrix, they may return through just these ATP synthase molecules. And when the hydrogens pass through this ATP synthase, the globular units will turn, and they will create ATP molecules from ADP and phosphates. A passage of each of the protons through the ATP synthase molecule creates one ATP molecule. So that is a process of oxidative phosphorylation. Just I mentioned it is actually so that the process that is responsible for formation of ATP. And acetyl coenzyme A formed through the beta oxidation of fatty acids and the degradation of the glucose is oxidized in the citric acid cycle to produce, in addition to carbon dioxide, so large quantities of the cofactors which are NADH and FADH, which give up 2 electrons, and these electrons will be transported through this chain from first to second and so on. Finally, the electrons join oxygen and the water is formed. And with each step, the hydrogens are carried to the intermembranous space. And the returning create, so as the protons pass down this electrochemical gradient, the energy differential in the proton motive force is transformed into the stable energy, high energy bond of ATP by the formation of the ATP from ADP and phosphate. And actually we have to know that each molecule of glucose will give us 36 molecules of ATP, but fatty acids, they produce more because they have more carbogen atoms. That's why they will create more amount of ATP. In some animals, also in newborn child, in their mitochondria there is instead of the ATP synthase, there are present thermogenin proteins that resemble ATP synthase, but they cannot generate ATP. And as the protons pass through the thermogenins to re-enter the matrix, the energy of the proton motive force is transformed into the heat, and it is this heat that awakens the animal from its state of hibernation. So that is the mitochondria.
Major function of most cells is the synthesis of proteins, either for use by the cell itself or to be exported for use elsewhere in the body. The primary components of the protein synthetic machinery of the cell are the ribosomes, rough endoplasmic reticulum or RER, and Golgi apparatus. We know that we have two types of endoplasmic reticulum, that is the smooth one and the rough, which is started with the ribosomes. The smooth endoplasmic reticulum, it functions in lipid and steroid synthesis, also in glycogen metabolism and detoxification of toxic substances, drugs, for example, if it takes place in liver. Also, as a function, other important function of the smooth endoplasmic reticulum, it is storage of calcium ions, which is very important in muscle tissue. If we are talking about the rough endoplasmic reticulum, RER, here we can see that it, for example, inside of this cell, we can see the electron micrograph representation of the rough endoplasmic reticulum that occupies most part of the cytoplasm. By more high magnification of the electron micrograph, we can see the individual ribosomes which are scattered on the surface of the membranes of rough endoplasmic reticulum. And we know that the function of RER, it is synthesis of proteins that are destined to be packaged either for storage within the cell or for the release into the extracellular space. So let's talk something about the ribosomes. The ribosomes are small structures, non-membranous organelles, and composed of the large subunit and small subunit. Each subunit includes ribosomal RNA and the proteins. The small subunit has binding sites for messenger RNA, then has P site for binding of peptidal transfer RNA. Here we can see transfer RNAs which will carry here their cargo, that is amino acids, which are needed for protein synthesis. Then A site, that is for binding of amino acid tRNA, and the exit site, E site, for exit of free tRNA, which gave its cargo to protein synthetic machinery. So let's go to now to talk about what is the protein synthesis and how it starts. We know that information about all of the proteins is written on DNA molecule inside of the nucleus. So the first step here is needed, that is transcription, or it is formation of the messenger RNA, which will carry information, the message from the DNA to the cytoplasm. The messenger RNA then leaves the nucleus and enters the cytoplasm, and here we can see the chain of messenger RNA, which contain sequence of codons, these three constitutive nucleotides. They form codons, and we know that each codon, it is code, it is information about one amino acid. So the next step we will see, it will be the process of translation. That means the translation of the language of codons into the language of amino acids in protein molecule, and it will be carried out by our ribosomes. Here we can see the ribosome. Here the P side of it, the A side and exit side we cannot see here, but what we can see here, that is transport RNA, which carried the next amino acid here to the place of protein synthesis, and peptidal transfer RNA, which carry the polypeptide chain. So we have to remember that proteins destined for the cytosol are manufactured within the cytosol on the free ribosomes, which together then will form polysomes. What is a polysome? You know that one messenger RNA may be translated by many ribosomes, and one messenger RNA together with the ribosomes, which read information, which translate this information into the protein molecule, that complex will call the polysome. So a little about how the proteins will be produced on RER. First of all, we have to recall that proteins on RER, they are needed to be packaged, and then they will follow the secretor pathway. They will be proteins of plasma membrane, for example, here we can see RER, then the Golgi apparatus. And so the proteins from RER will go in three ways. First, they will incorporate it into the cell membrane, or they will go outside of the cell, inside of the secretory vesicles, or they will be incorporated into the lysosome. They will be lysosomal enzymes. But the common feature of these proteins, which are going to be produced inside of RER, it is a presence of signal peptide, that is signal sequence. Here we can see it. That is a cistern of RER, and these are the ribosomes which joined to each other via messenger RNA. That is thread, it is a messenger RNA. So the signaling peptide, here we can see. The signal peptide then inside of the cytosol will be recognized by signal recognition particle, that is this one, number 4 here. And this SRP, signal recognition particle, binds to the signal peptide. And when it binds here, the protein synthesis here ceases, it stops. And the ribosome, messenger RNA, and SRP complex, all of these complex will be directed towards the RER. The membrane of RER have the integral proteins, that is signal recognition particle receptor, Then the ribosome receptor protein, that is riboforin first and second, which will recognize the large subunit of ribosome. And it also has pore protein, here number 6, which is formed by Sec61 protein. That will form pore here. And when all of this complex binds to the surface of RER, this signal sequence will be translocated and will enter the cistern of RER. Then the signal peptide will be cleaved off by the enzyme signal peptidase, that is black point here. That enzyme will break signal peptide, and the process of translation will continue till the protein produce the nascent protein. It enters, you see here, it enters the cistern of RER, and the protein synthesis will finish when the stop codon will be reached on the messenger RNA. And then it will give information that the protein synthesis finished, and all of the complex components, the ribosomal subunits, messenger RNA, they will detach from here. And the protein now will be translated. Now the protein is inside of RER. It undergo proper folding. It may undergo initial stage of segregation, glycosylation, and other needed steps of modifications. Then the completed proteins will be packaged into transfer vesicles to leave the RER and be transported to the Golgi apparatus for further post-translational modification and final packaging. But here I want you to pay attention at some point the process of formation or budding of vesicles from one organelle or from one cistern, and merging them with the other is aided by specific code proteins. Here we can see the code proteins, for example, this one, which is colored by blue color. And there are 5 major types of proteinaceous codes with some subtypes, some that cells use to accomplish these goals. These are cotamer proteins, COP1, COP2, clathrin, retromer, caveolin, and each code is used to encourage a specific type of transport. The code proteins also help vesicles to reach the proper destination to the target. At the site of future vesicle formation, these proteins accumulate. Here we can see they accumulate, attach to the membrane, draw out the vesicle, and coat its cytosolic surface. As the coated vesicle reaches the membrane of its target, here, at this site, here what we will see, when it reaches the target, it will lose its coat and fuse with the target membrane. And the delivered cargo then entered into the next organelle, next cistern. Here we can see endoplasmic reticulum and the Golgi apparatus. And here we have to understand something, some point. That's why I want to pay your attention here. Let's imagine that these vesicles carry cargo from ER to Golgi apparatus. After carrying to the Golgi apparatus, here they will be examined. And if here that vesicles which carried that cargo, if that cargo contains an escaped ER, endoplasmic reticulum resident proteins, which carried here mistakenly, on that case, that protein is returned to endoplasmic reticulum via vesicles which will be covered by COP1 proteins. And if in the first case, this transport is called anterograde transport. So on next case, which means returning of resident proteins to ER, this transport will be called retrograde transport. Because it will take place in opposite direction. What is a Golgi apparatus? It is composed of series of flattened, slightly curved membrane-bounded cisterns, known as faces, which do not quite contact each other, as we saw in rough endoplasmic reticulum. That's why all of the cargo will be transported via specific via specific transport vesicles. They will arise from one cistern, as I told you previously, and by that way this cargo will be delivered to next and next cisterns. The Golgi apparatus, it includes some parts. Let's look at them. First of all, here we have to mention the green colored zinc cistern. Actually part of rough endoplasmic reticulum, but it's devoid of ribosomes. It is called transitional endoplasmic reticulum. After, we can see here components of Golgi apparatus. That will be first component, which is called cis-Golgi network or proximal vesicular tubular network. Next component, it is called cis phase, then intermediate phase, then trans phase, and finally we can see this network, which composed of tubules and vesicles, and it is called trans-Golgi network or distal tubular vesicular network. So these are the components of the Golgi apparatus. Proteins that leave the transitional endoplasmic reticulum, here they came from endoplasmic reticulum, and they have to go to Golgi complex. How they will go there? So the proteins leave the transitional endoplasmic reticulum. They packaged in small transport vesicles whose membrane derived from endoplasmic reticulum and is covered by COP2 protein, that is coat protein. These COP2 coated vesicles travel to the vesicular tubular cluster, that is cis-Golgi network. Here they lose their COP2 coat, and fuse with it. The proteins are passed to the various phases of the Golgi apparatus, again via COP1 coated vesicles. Here we can see them. Where they are modified, here inside of the Golgi apparatus, they will be modified. They undergo some changing, the glycosylation, phosphorylation, they may acquire three-dimensional structure and other needed modifications. Then finally they reach trans-Golgi network. In trans-Golgi network, they undergo sorting and final packaging to be sent to the place of the address. And here we have to know that here from the trans-Golgi network, they sent to one of three places. Here we can see three kinds of vesicles which arise from trans-Golgi network. What are those vesicles? First of all, the secretory vesicles. The secretory vesicles, they contain the product of the cell, and they will be temporarily stored under the cell membrane, and when the cell will receive signal, they will release their content outside. So they need some signal. That's why this process will called discontinuous exocytosis. And here we can see formation of the lysosomes. The lysosomal enzymes will enter into these vesicles. These vesicles, some authors call primary lysosomes, or others call the transport vesicles which carry lysosomal enzymes. Or in some books you may read, you may see that there are written that they are late endosomes. In any case, these are vesicles which carry lysosomal enzymes, remember. And in both of them, we can see that they are covered by the clathrin protein. Clathrin, as we know, that is one of the coat proteins. So both of them, secretory vesicle and lysosomes, will be covered by clathrin protein triskelions, as we can see here. Then they will shed off their clathrin coat, and finally we will have our primary lysosome or transport vesicle which carry lysosomal enzyme. Finally, the components of the cell membrane will be carried here to its place of destination via specific vesicles which will constitute the constitutive secretion or continuous exocytosis, which do not need any signal, and it continuously taking place to replace the components of the cell membrane or the plasma membrane to their place. Or they may discharge outside, and that will be the components of extracellular matrix. So we have here two pathways, the constitutive pathway and the regulatory pathway, that will be for the secretory vesicles. So let's go to the next compartment of the cell, that will be the endosomal compartment. Is membranous compartment located near the plasma lemma, whose membrane possesses ATP-driven H pumps that acidify its lumen. This set of tubules and vesicles located deeper in the cytoplasm near the Golgi apparatus, and they help to prepare its contents for eventual destruction by the lysosomes. So what we have to pay attention, no matter that is an endosome or lysosome, in the membrane we will have the H pumps which will carry H protons inside the organelle. By the way, it will acidify the content, and the acid environment is very important, it's very necessary for the activation of the lysosomal enzymes. We know that pinocytic vesicles lose their clathrin coat and fuse with the early endosome. So here we can see the early endosome, a membranous compartment located near the cell membrane, and as we know, in the membrane of endosome we will have H pump. So the early endosome, inside of it, the pH will be approximately 6, will drop to the 6. Early endosome has central vacuolar region and the peripheral tubular regions. When these vesicles which carry cargo, the result of pinocytosis, these vesicles will fuse with these early endosomes. Inside of the endosome, under the influence of acid environment, the ligands will dissociate with the receptors. The receptors will return via recycling endosomes, will return to the cell membrane for reusing, and the ligands will remain inside of the endosome. By continuous pumping H protons inside of the organelle, it will lead to formation from this early endosome the multivesicular bodies by invagination of the membrane which surround the organelle. Multivesicular, it means that the organelle which include many vesicles. It also contains, as we know, the ligands which wait their turn to be destroyed by the lysosomal enzymes. So finally the late endosomes are formed. That is another membranous compartment located at a deeper level within the cytoplasm. The H pumps in the late endosomal membrane further acidify the lumen of this organelle, which continues to digest, and which is needed for digestion here. But what will digest these substances? Here we can see that from Golgi apparatus the vesicles are transported, and these vesicles contain the lysosomal enzymes which are needed for the digestion. Then they will fuse with late endosome. Finally, we will have final lysosome or secondary lysosome, inside of which these ligands will undergo digestion. Then we are coming to lysosomes. The lysosomes, this implies the name lyso, it means cut or digest. So that these are the organelles which house a high amount of hydrolytic enzymes that function at the low pH. It will be pH 5. That is achieved, as we know, that by the presence of H pumps on the membrane. Lysosomes degrade various substances whose useful components are released into the cytoplasm, and whereas the indigestible substances will remain enclosed by the lysosomal membrane, and the organelle finally, after digestion is completed, this lysosome organelle will become known as residual body. The lysosomal proteins, we know that they are produced in Golgi complex. We'll recall something, we will emphasize something here which we need to know for understanding the lysosomes. Here we are looking at the RER. And you know that the proteins which destined to enter the lysosomes, they are produced in RER. But these proteins, these enzymes, lysosomal enzymes, they need some additional group. This group will called mannose 6-phosphate group. Here, that is by blue colored, we can see the additional group for lysosomal hydrolase. That is lysosomal hydrolase precursor. After it binds mannose 6-phosphate group, it will form this lysosomal hydrolase, which transported to Golgi apparatus, and in trans-Golgi network, the membrane of trans-Golgi network, it contains the receptor, mannose 6-phosphate receptor. So the mannose 6-phosphate receptor will bind the mannose 6-phosphate group on each of these lysosomal enzyme, and all of the complex will be covered by coat protein, that is clathrin coat. Now we know it, and this clathrin coat will draw out the vesicle from here. That will be our transport vesicle, which contain lysosomal enzyme, or as I told you previously, we can call it the primary lysosome, that because it contains the non-active enzymes. Then it comes to the late endosome. It fuses with it, and in the acid environment of late endosome, the receptors will dissociate from the mannose 6-phosphate groups, and the receptor will bud off from here in vesicle, and it will return. So the receptors return to the Golgi apparatus for reusing. But the mannose 6-phosphate groups, together with these enzymes, they remain inside. Then the phosphate will removal of phosphate. Here we are looking. And finally, the lysosomal hydrolase will be activated, that is mature lysosomal enzyme. So here we looked at the fusion of primary lysosome with the late endosome. But we have to know that the lysosomes, that here the number 9, the final lysosome or secondary lysosome, may be formed by fusion of not just the first way that we can see here, the pinocytosis, endosomes, late endosome, and primary lysosome. But there may be possible through two additional ways. So first of all, this is heterophagosome. So substances destined for degradation within lysosomes reach these organelles in one of three ways. That is through late endosome, or heterophagosome, the cell swallowed some bacteria, for example, that is formed heterophagosome, or via autophagosomes. What are the autophagosomes? They contain the components of the cell which is no longer needed for it. So the smooth endoplasmic reticulum membrane will surround this organelle. It may be mitochondria or any other organelle which is not needed for the cell. It is surrounded by smooth endoplasmic reticulum membrane, so the autophagosome is formed. It also will fuse with the primary lysosome. Finally, the secondary lysosomes will be formed, and the process of digestion will take place. What we have to know also from about the lysosomes, that there are some diseases which called lysosomal storage disorders. In these disorders, the individuals, they suffer from deficiency or hereditary deficiency of certain lysosomal enzymes. As we know that lysosomes include so much hydrolytic enzymes, if one of them will the cell will not produce any of them, so the cell will be incapable of completely degrading that macromolecules which need that enzyme, and that macromolecules will not be degrading into soluble byproducts, and these intermediates will accumulate inside of the lysosomes, amassed within the lysosomes, and the size of lysosomes increases sufficiently to interfere with the abilities of these cells to perform their function.
Here we are looking at the lysosomal storage diseases. So in the normal cell, which have complete lysosomal enzymes, there is normal metabolism is possible, but if there is enzyme deficiency, it leads to storage of unbreakable components that need the missing enzyme. An amount of some diseases characterized in this group, lack of some lysosomal enzymes cause the storage of some metabolites in cell. Here we can see them, for example, Gaucher disease, and for example in Tay-Sachs disease defined in neurons because of lack of lysosomal galactosidase, hexoaminidase, and accumulation of residual bodies as concentric lamella, and results in dysfunctioning of the neuron. This was first defined in 1881. And then here we are looking at the disease type, that is glycogenosis, and this is Pompe's disease. For example, here the deficiency of lysosomal glycosidase, and metabolic buildup, it will be glycogen. Because of the deficiency of the glycosidase, glycogen will be stored. Another disease is sphingolipidosis, that is related to deficiency of the gangliosides, and here also will accumulation those components. And in sphingolipidosis deficiency of the gangliosidosis, that is a disease name, and missing enzyme is sphingomyelinase, which will lead to accumulation of the sphingomyelin and so on. So these are the major lysosomal storage diseases. Now we come to the proteasomes. Proteasomes, as implies the name, these are organelles which are designed to degrade proteins. But these proteins which are needed to be degraded within the proteasomes, they are endogenous proteins. It means that proteins which are produced inside of the cell itself, they will go to proteasomes for degradation. But which proteins will be degraded? Actually, those which are spent, which are no longer needed for the cells, or they maybe have, these proteins have some problems, and they are misfolded proteins, or they are antigen proteins of the antigens, some viral proteins that also produced within the cell itself. And so it means that proteasomes, they provide quality control of the cell, and proteins which are needed to be degraded within the proteasomes, they will enter the proteasome. Proteasome has a barrel shape. Here the regulatory particles, this one, and this is a central barrel which include 2 alpha and 2 beta subunits. The beta subunits have the protease activity, the enzymatic activity, and they will break those proteins, but the proteins will be broken down not till the end products, not till the amino acids, but till the peptides, which include 7 to 8 amino acids. So these are peptides which are result of protein degradation inside of the proteasomes. So protein degradation occurs on default of folding of proteins, and we have to know that the proteins which needed to be degraded within proteasome, they have to be marked. And the marking here, that is a ubiquitin molecules. The ubiquitins will be recognized by the recognition particle of the proteasome only after the protein can be dislocated into the barrel. So the ubiquitin is needed to mark that protein for needed to be degradation. And also here we can see transmission electron micrograph representations, images of proteasomes that were made in different cell types. These are proteasomes, and these are large protein complexes with the hollow cylindrical shape. Now we are coming to the peroxisomes. Peroxisomes, these are these organelles. They are membrane-bounded organelles. They contain different kinds of enzymes. There are urate oxidases, catalases, deamino acid oxidases. The function of the peroxisomes we can subdivide into anabolic functions and catabolic. Anabolic, it's related to production of the cholesterol molecules, and but the catabolism activity is catabolism of long-chain fatty acids to produce acetyl coenzyme A. And another function of it, that is the metabolism of hydrogen peroxide. Hydrogen peroxide is formed as a result of beta oxidation of fatty acids with long chains, and as a result of those reactions the hydrogen peroxide is formed. And hydrogen peroxide is needed for detoxification of toxic substances like ethanol. But also hydrogen peroxide has antibacterial activity. But excess of hydrogen peroxide is destroyed by catalase, which is present also in peroxisomes. Peroxisomal enzymes manufactured in cytosol, polyribosomes, some membrane proteins in rough endoplasmic reticulum. So the proteins which needed for peroxisomes, they produce mainly on the free ribosomes. And peroxisomes also, like mitochondria, they increase in number by fission, by subdivision into two. And they do not have their own genetical material. We also have to know it. And then we are going to peroxisomes appearance under the electron microscope here. Inside of the peroxisomes we have crystals of urate oxidase, and that is the here we can see the immunohistochemical staining of peroxisomes. And here also these structures are peroxisomes, which include this crystalloid structure. The electron microscopic appearance of peroxisomes in rat liver. In the liver cells, we are looking at the peroxisomes. And in this case, inside of the cell, the transmission electron micrograph of the cell, we can see the organelles. These are mitochondria. Then we are looking at the Golgi apparatus, cisterns of Golgi apparatus, then rough endoplasmic reticulum, and finally peroxisomes. So that's all for today. See you on next lectures.